understanding of life science, and improving healthcare. In vaccine design, bioinformatics helps in predicting antigenic protein, modeling immune responses, and selecting vaccine candidates. It helps expedite vaccine development, improve efficacy, and address emerging infectious disease effectiveness. Now discussing about proteomics and genomics. Proteomics and genomics provide extensive biological data, while bioinformatics offers tools to analyze and interpret this data, enabling insights into genes, proteins, pathways, and structure. Together, they drive advances in biology, medicines, and molecular understanding. In comparative genomics, bioinformatics facilitate the comparison of genomes across species, revealing evolutionary relationship and functions. Structural genomics helps predicting the three-dimensional structures of proteins encoded by genes, aids in understanding their functions and interactions. In both genomics and proteomics, bioinformatics accelerates research, enhances data interpretation, and facilitates a deeper understanding of biological processes, ultimately contributing to advances to medicine and biology and basic science. Bioinformatics enables the use of systems biology to model, analyze, and simulate complex biological systems. It aids in understanding how genes, proteins, and molecules interact within cells, leading to application in disease research, drug discovery, and personalized medicines. So, under the guidance of Dr. Bhasha Javed sir and his academic and industrial experience of almost 10 years from various reputed institutes, including Helix Infosystem Limited, Noida, Bioinformatics Institute of India, Noida, Indian Biological Sciences and Research Institute, Noida, SPS Education Group, Bareilly, and now currently in Bioinformatics Technologies of India. BTI has achieved numerous milestones and had some healthy collaborations for almost 8 years till now with highly reputed institutes over time, including Noida Institute of Engineering and Technology, NIT, Noida, Pine Biotech, USA, and IFTM University, Moradabad. At Bioinformatics Technologies of India, we offer industry-leading bioinformatics training that caters to both beginners and seasoned professionals. Our mission is crystal clear: to provide a platform for individuals, professionals, R&D organizations, pharmaceuticals, and software companies to harness the immense potential of Indian bioinformatics. You can visit the official website of Bioinformatics Technologies of India for further details by scanning the QR code in the end. So now let me help you decide why you should choose Bioinformatics Technologies of India. Bioinformatics Technologies of India have always been dedicated with extensive curriculum, hands-on experience, expert facility, network opportunities, flexible learning, career-oriented program, and industry collaboration. We believe in do and learn. So don't miss the opportunity to revolutionize the field of biology through bioinformatics. Enroll in our training program today and embark a gearing up journey of innovation discovery. Unlock your potential and shape the future with bioinformatics training from Bioinformatics Technologies of India. Now, without any further delay, I would like to call upon today's speaker, Sanjay, to continue with the webinar.
Thank you, Tushar.
Over to you, sir.
So, can you see my screen now?
Yes, it is visible.
Yes, sir.
Okay. Put it in the presentation mode. So you're able to see the full screen, right?
Title is there.
Sorry, come again. Title is there.
Right.
Okay.
Yeah.
Yeah, awesome. So, hi everyone. I'm Sanjay. I'm a bioinformatician and a DevOps working in Germany. So my background is I did my bachelor's in bioinformatics in India, master's in bioinformatics in Germany, and then PhD in Germany as well in the field of bioinformatics and so on and so forth. But less of that story, but more of today's webinar. Yeah, thank you everyone for joining. So today's webinar is titled Unlocking the Power of Bioinformatics, Exploring the Current Trends and Cutting Edge Tools. So I see there will be a brief overlap between what I'm going to talk and what Tushar briefly mentioned about services in BTI and bioinformatics. So bear with me for a couple of minutes, and then we'll go into the other topics and such. Okay. So a brief overview of bioinformatics. So by now most of you know that it's an interdisciplinary field which combines biology and computer science, right? But also in addition to these fields, you also have the domains such as statistics and mathematics and so on and so forth, to analyze and interpret the biological data. And usually bioinformatics plays a crucial role in understanding complex biological processes, in identifying genetic variations, protein structure and function relationships, and in disease mechanisms and so on and so forth. Usually bioinformatics, or a person who is doing bioinformatics is called a bioinformatician. And bioinformaticians these days are mostly known as bioinformagicians, simply because a bioinformatician should be capable of handling and managing big data, collaborating and communicating with several stakeholders in several areas and so on, should be capable of doing problem solving, and should be equipped in designing, developing new algorithms, data structures, and tools to analyze these data and perform high throughput computational analysis, and so on and so forth. So that's why, in addition to these, bioinformaticians are also required these days to be well versed with programming languages, computer science, Linux operating systems, in maintaining HPC infrastructures, data storage, and so on and so forth. So that's typically why bioinformatician is like a one-man army, a one-person army who can deliver a lot of things. Yeah. So that's basically a very brief overview of bioinformatics and a bioinformatician in general. So most of us know what is the central dogma of molecular biology. Yeah. So which is basically DNA transcription to RNA and then translation to protein. Yeah. So when we apply that onto the bioinformatics analysis, from molecular biology to bioinformatics transition, so we do that in different areas of research, or different areas of different domains inside bioinformatics, which is genomics and epigenomics, where you study, where you can, or where one can study the DNA sequence and associated heritable biochemical modifications and so on and so forth. For example, histone modifications or methylome data analysis and so on. Yeah. And then you have, for studying RNA, you have the RNA molecules, which is the field called transcriptomics. And then you have something called proteomics, where you study the proteins present inside a sample. And finally, studying the small molecules are known as metabolites, otherwise known as metabolites, or in the field of metabolomics. So the ultimate goal is basically to understand the complex relationship between the geno to pheno, yeah, on a global scale, and bringing data to life through these range of multiple processes. Yeah. So omics, we have heard a lot of about, we heard a lot about it. And so what can it basically tell us? So imagine you have a patient data, a sample from a patient, and you want to study the functional. So this patient data is associated with a cancer. Yeah. You have a reference data and you have a patient data. So in patient data, you want to analyze a particular mutation or something that is leading to carcinogenic reactions and carcinogenesis and so on and so forth. So you can, for example, use genomics and study using whole genome sequencing or whole exome sequencing. You can perform various types of data analysis. It could be point mutation, or it could be studying of SNPs. It could be studying of copy number variation or structural variation and so on and so forth. Similarly, you have other technologies where you can use transcriptomics, single cell transcriptomics, spatial transcriptomics, and so on. Whereas, if you want to study a bit more detailed of on or off mechanisms, you can also study the epigenomics parts. You can do bisulfite sequencing and chip sequencing to explore the methylome data and the histone modifications that's happening inside, and so on. So at the end of the day, the goal is basically to further our understanding in and coming closer to solve cancer related diseases, or personalized medicine, and so on and so forth. Yeah. So genomics or sequencing. So the evolution of sequencing technologies, yeah, the basically it started in 1977. Yeah. The first, that's called the first gen sequencing, which uses chain terminating ddNTPs through DNA polymerase during in vitro DNA replication and so on. And this first gen sequencing produces relatively long read lengths, up to 1,000 base pairs, otherwise known as 1 kilobyte kilobase, and higher per base accuracy, that is approximately 99.99% accuracy. However, the first generation sequencing technologies usually suffer from high cost and low throughput that call for novel technologies delivering fast, inexpensive, and accurate solutions. So this led to the arise of second generation sequencing, otherwise known as the next generation sequencing. So this dramatically improved the speed and scalability of genome sequencing through various methods. During this period, several commercial products were made available from 454, solid sequencing, Solexa, Ion Torrent, Illumina, and whatnot. But but due to several political and could not survive, for example, 454 left the field of next generation sequencing. However, a couple of companies survived throughout and made milestone contributions by improving the throughput that first generation sequencing could not offer. So these next generation sequencing basically outweighs the first generation sequencing, otherwise known as Sanger sequencing techniques, in higher speed and throughput, easier gene library construction, higher parallelism, and so on and so forth. Yeah. However, this second generation or next generation sequencing also basically suffers from short read lengths. As you can see, it can produce reads up to 500 bases or something like that. Yeah, and that lead to at least 10% fold less accuracy when compared to the first generation of Sanger sequencing. And these short read methods cannot well capture structural variants, repetitive elements, the higher low GC content, or sequences with multiple homologous elements in the genome, and so on. So this led to the arise of this third generation sequencing, otherwise known as TGS. This revolutionized the sequencing technological field in the last span of from 2012, yeah, something like that, 2012, 2013. This has been going on. These new technologies by PacBio or Pacific Biosciences, Oxford Nanopore, they use something called an SMRT technique, single molecule real-time sequencing technique, where they put in more nucleotides through for the DNA polymerase to happen, and so on and so forth. And the sequencer is equipped enough to handle all of those things, unlike the previous generation sequencers. Yeah, and this technique that allows for the or enables the production of long read sequencing with low alignment and mapping errors during library construction. And these new techniques can detect hundreds of kilobases in one continuous read, and which is the major advantage when compared to the previous generations. And such sequences are ultra long reads, can span anywhere between 300 kilobases or even up to 1 million base pair in certain use cases, in certain cases. Yeah. And yeah, one can apply these sequencing technologies to study various outcomes, and basically your imagination is the end there. So comparison between short reads or long reads is that short reads can capture the majority of the genetic variations, whereas long read sequencing allows the detection of complex structural variants, as well as that may be difficult to detect in short read sequences. Yeah, so this is the major advantage when we are doing long read sequencing and so on. So with the development of technology, we also have issues these days when it comes to data, when it comes to other things as well. So first topic would be cost wise. So we could see, so basically Moore's law indicates that for every 2 years there is a twice in growth, right? So here we see the complete contrast, that here Moore's law indicates that prices of computing decline at approximately 2 to 3% every month. So the cost of DNA sequencing per genome may amount to the fastest price decline in history that we have seen so far. Yeah. So with this, cost going down for doing human genome sequencing or any sort of DNA sequencing at any depth, there is a extraordinary increase in an exponential way in data getting deposited, the sequence data getting deposited in databases like European Nucleotide Archive and Sequence Read archives and so on. Yeah, so in the last decade, we see that public databases like SRA, for example, contains approximately 25.6 petabases, which shows that more than 18 to 20 petabytes of data is sequencing data has been stored. Yeah, this 25.6 petabase pairs originates from 14.8 million publicly available sequencing runs. Yeah, you also have publicly available ones and also controlled access reads, which are different. Yeah, some of them can be accessed by everyone in public. And these 14.8 million publicly available runs averages to 1.7 gigabase pairs per run, which is approximately zero point 0.8 or 700 to 900 megabytes of storage per run. Yeah, so we can see that as the cost of sequencing goes down, the amount of data that one needs to analyze to make efficient analysis out of this goes scales up really quickly, whereas our technology to understand that is not up to the par. Yeah. So here we can see that genomics is shown as a genomics is basically a big data science in this in 2023 that will get just much bigger very soon. Yeah, but it is yet to be known whether the needs of genomics will exceed other big data domains. For example, here 4 data domains have been shown by the authors. One is astronomy, Twitter, and YouTube, and so on. And projecting this to the year 2025, the authors of this paper compared genomics with these 3 major data generating fields. Yeah. There, the estimates they made that shows that genomics is basically a four-headed beast when you see that it requires up to 2 to 4 petabytes per year of storage. Yeah, and then we can produce up to 1 zeta bases per year. So that's why they coin the term four-headed beast. And it is either on par with or the most demanding domains analyzed here regarding data acquisition, storage, distribution, and analysis, and so on. And they also discuss the aspects of new technologies that will be needed to be developed to rise and meet the computational challenges of genomics in the near future. And basically, now is the time for us to have a concerted community wide planning for the genomical challenges for the next decade. Otherwise, it'll become really crazy in matter of few years. Yeah, and basically we are drowning with data. So with that, let's have a quick look on. Let's take a long walk to genomics, the basically the history and the current approaches. Yeah. So genomes represent the starting point of genetic studies. So since the discovery of DNA structure in 50s, scientists have devoted great efforts in determining their sequences in an exact way, in a precise way. How can we go from DNA structure to the sequence? So this timeline here shows the major milestones achieved in genome assembly. Basically from the beginning of sequencing era in 1965 to the ongoing large-scale genome projects in 2020s. Yeah. So each genome or a genome project shown here is placed under different color-coded backgrounds. So you can see that one in red are from the early sequencing methods, and one in the yellow shows the Sanger sequencing based shotgun sequencing methods, and the green is the second generation sequencing methods, and finally you have the third generation sequencing methods over here in the blue. So in 1965, the first one they categorized was or sequenced and assembled was the 76 base long tRNA sample from yeast, Saccharomyces cerevisiae, was sequenced, and in when we jump from there to 1975, a decade later, Sanger sequencing started. Sanger and Carlson developed the plus and minus method, where they use the ddNTPs and so on for DNA sequencing, and this was used to determine short regions in couple of phage single-stranded DNAs that we see here in 1975. Moving there again a decade later, we see in 1984, yeah, in that period, around that period, we see that genome of Epstein-Barr virus was sequenced, which comprised approximately 180,000 base pairs, which was determined using again shotgun sequencing methods. And starting from the 1980s, many full genome sequencing projects were launched and succeeded. And in the 90s, there was this technological breakthrough from first generation sequencing combined with the adaptation of industrial processes, which increased the throughput and reduced sequencing errors and so on. So these advancements were further accelerated by the Human Genome Project, which started sometime in 1990. The aim of the project was to produce genetic maps and physical maps, and finally the complete sequence of human chromosomes, all the 23 chromosomes. The work almost cost approximately $2.8 billion, yeah, over the period of 13 years. And in this period of 13 years, there were a huge technological advancement within the next generation sequencing domain or the second generation sequencing stage. So this human genome resequencing efforts that started later to become more affordable simply because several companies realized that yes, sequencing could be a profitable business. And therefore, the market competition gave birth basically to plethora of sequencing technologies that are currently there now and collectively referred to as next generation sequencing. Yeah. This further gave rise to the 1000 Genome Project and then the 10,000 Genome Project. So here in 1000 Genome Project in 2000, somewhere in 2008, 9, and then it also started with this 10K Genome Project by different people, and then the Zebrafish Genome Project. And in 2015, late 2015, there was this all species genome project where they wanted to for every living organism in earth they wanted to sequence, they wanted to collect the data, they wanted to sequence that. And yeah, and more recently there are the projects such as this vertebrate genome project, which is trying to sequence all 71,000 or 71 million vertebrates that are present. And then you have these projects such as Tara Oceans project, which wants to sample, this is a metagenomic project, so which wants to sample every ocean that is present and every water that is available to to sequence, to identify the different species that is present in these water bodies and so on and so forth. Yeah. And the ultimate goal was to sequence and catalog and categorize all species in the world, covering space, earth and water, all directions. So that's what's happening on the global scale when we talk about sequencing efforts. Yeah. So we have seen the advancements in sequencing technology, which basically led to rapid growth of sequence information. So these NGS data is likely to contain, actually most likely to contain clinically significant important information, but whereas our ability to mine these effectively are largely limited, simply because searching these databases are not possible in a reasonable computational time. So therefore, large requirements for tools to query these databases are required, simply because BLAST-like algorithms, most of us would have used BLAST, yeah, in during bachelor's, master's, during PhD journey, or anywhere whenever we want to do some sequence analysis. If we have a random sequence and if we want to understand where the sequence originates or what species and organism contains these sequences, yeah, we would have used BLAST-like algorithms. But unfortunately, BLAST-like algorithms do not scale very well with the amount of data that we have deposited in databases like ENA and SRA, because BLAST-like databases, as the database grow, the complexity of BLAST to compute and map to the queries, the reference is quite very weak in that place, and it's very slow and it cannot scale very well. Therefore, in recent years, various alignment-free tools such as sequence bloom trees, COBS, Mantis, Chemtrix, Metaprofi, etc., were introduced, which uses several data structures such as bloom filters, cuckoo filters, quotient filters, bloom trees, and whatnot. So these are something called membership query data structure. So for example, when we have a search term and we take this search term and go to Google and then ask Google what is bioinformatics. So Google behind the picture basically indexes all the documents that we have or that they have in possession. And as soon as when we enter the word what is or search for this term what is bioinformatics, Google will take this and then looks through this index and identifies the documents that it has and then displays all those documents for us so that we can further proceed. And some of these technologies, some of these data structures are behind these search engines, information retrieval, and so on and so forth. So bioinformatics is borrowing existing algorithms or existing data structures and tweaking them, optimizing them to utilize efficiently for doing sequence searches and sequence analysis through bioinformatics for the ever-growing data sets like NGS and third-generation sequencing and so on. So what is the ultimate goal of such tools? So it is basically to index comprehensive collections of both amino acid and nucleotide sequences and enable the searching of large collections of sequencing experiments for any given query. It could be to analyze variants, it could be for taxonomic identification, annotation, classification, categorization, and whatnots, and binning, and so on and so forth. So, and also, it is very, very crucial that we should be able to achieve this with low compute requirements and orders of magnitude faster, and further enable query of the amino acid index using nucleotide sequencing. So, for example, if you want to study a variant, right, you can simply index a database called card db, which contains the resistant information of all bacterial genomes and stuff like that. So you can now imagine that you have a sample. It's a metagenomic sample which you obtain from somewhere, and you want to see you have sequenced it, and now you can use this resistance database that was indexed through one of these tools, and then just use the nucleotide sequence to query this amino acid index database, and then in turn you will be getting the information about what sort of variants that it is present, what sort of annotations or what species it has, and so on and so forth. Yeah. So these are some of the goals that these alignment-free tools in sequencing analysis these days are trying to solve or trying to address with limited or restricted compute requirements, and trying to scale as fast and as big as possible. So a quick example of that is that whenever people are developing k-mer indexing tools or indexing tools, they try to benchmark against something called a de facto benchmark data set, which contains sequencing data from samples like blood, brain, and breast samples. And this is an RNA sequence data set. And here what I'm showing is, so this data set contains 650 samples, the small data set, out of 2,585 samples. And the number of k-mers when we set k equals to 21 is approximately 8.8 billion k-mers. Yeah. So k-mers are nothing but short stretch of sequences, short stretch of overlapping sequences. So when they use data structure like Bloom filter, they predetermine a Bloom filter size called 2, and they set it to 2 billion. And to index this, approximately one would require 151 or 602 gigabytes of storage. So this table here shows that in by using only 64 cores and around 12 gigs or 2.4 gigs of memory, one can index this 650 samples containing 8.8 billion k-mers in less than 25 minutes. Yeah. And the total disk size at the end of the day is only 17.6 gigabytes. So this shows that one can index efficiently the data sets that's available, terabytes or petabytes of data that is available in databases, publicly accessible databases like EN and SRA, and where we can use these algorithms to answer several questions that we have. And they've also shown through a quick example of the querying process, and that shows approximately in how much seconds they were able to query 1,000 transcripts which are randomly selected from these experiments, and that shows that within in 33 seconds or in 22 seconds they were able to query 1,000 sequences. Yeah, this shows how much the technology has developed, and still what is required further to advance ourselves in sequencing analysis and so on. So that's for genomics. So let's quickly jump to proteomics or structural bioinformatics. So there is a huge gap between available protein structures and protein sequences, right? So in 1958, we determined first protein structure through different technologies and different methods like NMR, X-ray crystallography, cryo electron microscopy, and so on and so forth. And as of now, in 2023, we see that PDB contains approximately 210,000 structures, whereas UniProt contains approximately 252 million sequences. So you see the lack of you see the gap between the protein sequence and the structure that is available now, that are experimentally resolved. So it calls for huge improvements and advancing the field in this area. So basically, this is a 50-year-old folding problem which tries to address three broad questions. So what is the physical code by which an amino acid sequence dictates a protein's native structure? Is it possible, and how fast the proteins are folding in its native form? And can we employ computer algorithms to predict the protein structures from their sequence? So this was the problem that was coined 50 to 60 years ago. Yeah, and after that we have now sequence to structure prediction algorithms and so on and so forth, especially CASP. CASP is basically a kind of an Olympics in protein structural field, where they try to assess and evaluate the state of the art methods that try to predict protein structures de novo, or reference based and homology based and so on and so forth. So to address this problem, recently in the last 2 years, Google's DeepMind came out with a solution called AlphaFold. So this AlphaFold is basically an end-to-end solution for the protein structure prediction problem. Basically, it takes, you submit a sequence, and it AlphaFold behind the picture uses several things such as a genetic database, pairing pairing matrices, and then experimentally resolve structural information and so on, and then builds a multiple sequence alignment. And along with that multiple sequence alignment and the pairing information, both of these are embedded into neural network architectures to obtain protein 3D models. Yeah, and these models are further energy minimized and optimized through amber force fields and so on. And at the end of the day, we obtain relaxed models along with the quality scores to assess these models. And in this example, we see that AlphaFold in the one of the CASP experiments or CASP analysis. So they show how better they can predict protein three-dimensional structures. For example, in this example, we can see when compared to the experiment structure, we see an RMSD value of 0.8 Armstrong. So the lower the RMSD, the better the protein structure, that the predicted protein structure is when compared against the experimental structure. However, there are limitations, of course, such as in some cases, the model accuracy for different models by AlphaFold is small, is low, and AlphaFold struggles in some places when it comes to protein complex predictions, protein-protein interactions, and so on. And sometimes it also is unreliable when it comes to intrinsically disordered proteins. And AlphaFold relies on co-evolutionary relations, that is through the structural database that it uses, or the genetic information that it uses through the databases that we provide for AlphaFold analysis and so on. Yeah, but this has led to plethora of research in this field, in this domain, and there are several newly developed algorithms, several newly developed neural network models available these days, that is trying to do a better job in depending on specific domains, and some are in generic, doing efficient predictions, for example Rosetta Fold or Open Fold and so on and so forth. Yeah. So later, AlphaFold and EBI, European Bioinformatics Institute, joined hands together and created an AlphaFold database. So this prediction problem is predicting protein structures is one of the fundamental grand challenges in protein structural field or structural bioinformatics field, even including structural biology. Yeah. So such structures can dramatically deepen our understanding of human health, disease, and our environment. So earlier we saw that we only had 210,000 structures experimentally resolved, and now with AlphaFold, we are able to theoretically or computationally solve almost 214 million protein structures, and AlphaFold has deposited all of them for public access in AlphaFold DB, which also includes 48 complex and complete proteomes, including human proteomes to date. So this is another paper, Alpha Missense, again a tool, AI tool by DeepMind, which was published, I think, a couple of months ago or last month. So this new AI tool classifies the effects of 71 million missense mutations. So missense mutations are nothing but variants or genetic mutations that can affect the function of human proteins. So it is just one amino acid change, and this one amino acid change can lead to cancer as well. Yeah, right. So these are classified as missense mutations. And classifying these missense mutations or variants is crucial for faster diagnosis and developing life-saving treatments and so on and so forth. So the AI behind Alpha Missense tool categorized approximately 89% of the 71 million possible missense mutations, in contrast to only 0.1% which have been confirmed by human experts. And this also shows that they have achieved a significantly high specificity or the accuracy through AU rock, where they show 94% of uh, ClinVar variants have been categorized correctly. So ClinVar is basically a database of clinical variants where it has been experimentally solved and curated, and where they categorize whether particular mutation is benign or pathogenic. Yeah, so basically, alpha missense works in a way where you submit an input sequence with a variant, and then it builds the structure context using the alpha fold, and then employs new protein language models, and then it trains the variants, and finally it says whether particular mutation is benign or pathogenic. Yeah. So, as promised in the title of the webinar, some of the current trends that I was briefly able to collect, and so that I can share with you. So the current field is single cell. One of the fields is single cell. So in the past, we had bulk omics, data analysis, and these all provided valuable insights into various cellular behavior and so on. Whereas today, through single cell, we can explore heterogeneity within the cellular populations, within the cell populations and so on. So you can study at individual cell level. So from single cell RNA sequencing to single cell proteomics, we can now uncover distinct cell subtypes, translational states, and the rare cell populations and so on. So imagine the potential that this can bring us when we want to study independent and intricate cellular mechanisms in identifying cell-specific therapeutic targets in personalized medicine and so on. Yeah. There is this another field where you combine multiple omics techniques from the genomics, transcriptomics, epigenomics, metabolomics, metagenomics, microbiomics. You can try to combine all sorts of multi-omics data and create a harmonious symphony of information there, and to gain a comprehensive overview in understanding biological processes, disease mechanisms, and the intricate interplay between different types of data that we have to address geno to pheno issues. Of course, you have AI and ML. We already saw a couple of examples where the techniques and the technology involved showed that it revolutionized the fields by automating tasks, identifying patterns, and predicting the outcomes, and so on. And from clustering problem to classification problem to feature selection and network analysis, AI can improve. AI can predict things faster. So crunching through these massive data sets that produced as a end result of cheaper sequencing these days, we can try to leverage the AI and ML and DL techniques to uncover the hidden pictures and get benefited from the data-driven decisions exclusively. And of course, you also have spatial omics. So spatial omics technology enables a deeper understanding of the cellular organizations and interactions within a tissue of interest. So this combines multiple fields and enables the identification of specific compartments or regions in a tissue with that are differentially expressed, and different transcripts or certain protein abundance, and so on and so forth. And finally, this gives us a deeper understanding of cellular interactions, tissue microenvironments, and disease specific alterations. Of course, you also have metagenomics and microbiomics, where you collect samples from environment and you sequence them and you analyze them to improve the taxonomic resolution that we currently have. And this field offers compositional and functional information and aids in improving and identifying novel viruses, proteins, and characterizing genomic diversity and function of bacteria, identifying taxonomy of different species, identifying new things, and metabolic pathways that differentiate disease states, and so on. And these are some of the current trends that's happening in the field of bioinformatics research. So, additionally, I would like to briefly touch upon a couple of topics, which I don't know how many people are familiar in India, or the people in the audience. So, here people talk about something called FAIR data. So, FAIR means findable, accessible, interoperable, and reusable. So, findability can be ensured by using a unique or persistent identifier with sufficient description of data and their characteristics in machine readable metadata form, and storage of data in archives or repositories. So, accessibility requires that this metadata are always available via standardized, universally implementable communication protocols, and so on, with clearly defined access conditions. Yeah. And interoperability means that using this metadata can be used and accessible and in a standard language, so that we can reuse them. That is the reusability part, which is the ultimate aim of anyone's research, right? So detailed description of characteristics according to the community standards with clear conditions enables one to reuse the research data that's already been developed for or produced for future endeavors. And it's also directly embedded into something called research data management. This is also a crucial topic in bioinformatics, and a bioinformatician is required these days to know about fair data and research data management at the end of the day. So the research data management basically refers to all the measures taken to ensure the quality of research data. So it covers all the phases of research data lifecycle, that is from planning to collecting, storing and analyzing, through to describing, archiving, sharing, and reusing any of these research data. So there are basically, it falls under two main objectives. One is the transparency it brings when you do this, and as well as avoiding duplicating work by enabling research data to be reused by everyone in the field. And that's why it is considered essential and a good practice to follow FAIR principles when it comes to data, and have a good planning when it comes to RDM of the data that you are working with. Also, the other current trends are bioinformatics workflow tools, so SnakeMake, Nextflow, Galaxy, Common Workflow Language, and there are several other workflow languages and tools are available these days. And these help you develop workflows that can be reused and reutilized on different data sets and different things. And this is one of the hot topics these days, especially Galaxy, which provides a UI platform for your data analysis, and so on and so forth. Yeah. And it's also 100% free if you use nationalized, continental wide available use Galaxy infrastructures. For example, in Europe, there is something called usegalaxy.eu, which has all tools that one would require. And they also provide the computation for free, so you can go upload your data set and then you can run sequence analysis. They also provide training materials called Galaxy Training Network, and where they also have workflows designed for you in the form of training. You can take those workflows and use those workflows to analyze the data, and there is a really, really large community uh, in the Galaxy ecosystem. So Galaxy is there is one for Europe, and there is one in Australia, and there is one in United States as well. So and it's completely open source. It's completely free. Anyone who wants to install for their local institute, they can also do that. Yeah. So the last topic that I would like to touch is virtual research environment. So virtual research environment is another topic where people are investing a huge amount of money, especially in terms of trusted research environment, meaning that you have some hospital data which is not supposed to be shared or which is not supposed to be leaked outside, and how can you efficiently analyze them without sharing or spilling the data outside, yeah, without violating any of the agreements. So VR is we VR is basically comprises of the Jupyter Notebook, RStudio, Select Gene, where you can perform sequence analysis and so on and so forth. Yeah, we can come on this topic if someone has any doubts. But
Sanjay, there is a
am I
question.
am I still audible?
Yeah, yeah. Yes, you are audible. And
Okay,
there is a
okay,
question from Muhammad
okay.
Abu
With
Hussein.
this, one second. With this, I would like to close the webinar. So these are the some of the current trends that's happening in the field of bioinformatics globally, and to learn and to deploy further things, to learn programming and to efficiently implement these kind of algorithms and develop data structures. Programming is a very fundamental. It's very fundamental when it comes to bioinformatics these days. And so BTI is offering a bioprogramming course. You can scan this QR code and you can participate in the course, and all the details relevant for the course is available through the QR code. You can also talk to Dr. Basha, and you can get additional information through BTI and so on and so forth. So with this, I'll stop sharing. Okay, I'll leave that. And if there are any questions, I'd be happy to answer.